Hi, my name is Maya Landeros,
I'm Michaela Cavazos.
And I'm Alondra Curiel.
and we're going to be going over the Chapter 5 and Chapter 6. What are the functions of helper T cells and cytotoxic T cells and cell-mediated immunity? Helper T cells help in the activation of macrophages, which helper T cells stimulate macrophages to enhance their ability to kill ingested microbes. Recruitment of leukocytes, where they secrete cytokines that attract and activate other immune cells to the site of infection. Activation of B cells, where CD4 plus T cells assist in the activation and differentiation of B cells to produce antibodies. Cytokine secretion, where they produce interleukins such as IL-2 that promote T cell proliferation and differentiation. Enhancing APC function, where helper T cells interact with antigen-presenting cells, APCs, to improve antigen processing and presentation. Cytotoxic T cells work in killing infected cells, where CD8 plus T cells recognize and destroy virus-infected or cancerous cells by inducing apoptosis, releasing cytotoxic molecules. They secrete preferin and granzymes, which create pores in infected cells and trigger cell death. Memory cell formation, where some CD8 plus T cells persist as memory cells, allowing a faster response upon future exposure to the same pathogen. What are the steps in the activation of T lymphocytes? Naive T lymphocytes recognize antigens presented by antigen presenting cells through the T cell receptor and CD4 CD8 co-receptors. The TCR specifically binds to the peptide MHC complex, providing the initial activation signal. However, full activation requires co-stimulation, where co-stimulatory molecules on APCs bind to CD28 on T cells. Without this second signal, T cells may become unresponsive or undergo apoptosis. Following activation, cytokine signaling, particularly interleukin 2 production by T cells, stimulates clonal expansion, leading to a rapid increase in antigen-specific T cells. These activated T cells then differentiate into effector cells such as helper T cells and cytotoxic T cells, as well as memory T cells. Effector T cells migrate from lymphoid organs to infection sites, where CD4 plus helper T cells secrete cytokines to enhance immune responses, while CD8 plus cytotoxic T cells directly kill infected cells. Their migration is guided by adhesion molecules and chemokines. Once the infection is resolved, most effector T cells undergo apoptosis, while a subset of memory T cells persists, enabling a rapid and robust response upon future encounters with the same antigen. What signals are needed to activate T lymphocytes? The signals needed are antigen recognition, co-stimulation, and cytokine signaling.
So the two pathways that are present are the preferrin granzyme pathway, which cytotoxic T cells recognize infected cells via TCR MHC class 1 interactions, and the cytotoxic T cells kill virus infected cells. And within the perform granzyme pathway, the cytotoxic cell releases perforin, which is a protein, thus creating holes within the infected cell membrane. CTLs recognize infected cells through the MHC class, and this forms an immune synapse where cytotoxic molecules are delivered directly to the cell in need. The serine protease granzyme then is entering the cell through the small pores made by the previous protein. These granzymes then go ahead and activate capsases, siding proteases that play a vital role in initiating apoptosis. The next pathway is the Fas ligand pathway. This interaction begins when CTLs express Fas ligand, which binds to Fas receptors CD95 on the target cell. This then activates capsase 8, which then triggers capsase 3, which will eventually result in apoptosis as well. Unlike the other pathway, there is no need for the protein or enzyme in order to induce cell death.
What cellular receptors and ligands are used to sense and respond to activate T lymphocytes? T cell receptors recognizes peptide antigens presented by MHC molecules on APCs. CD4, CD8 co-receptors bind to MHC class II or MHC class I molecules to assist TCR signaling. CD3 and zeta chains signal transduction molecules associated with the TCR complex, adhesion molecules such as integrins LFA1 and ICAM1 strengthen the binding of T cells to APCs. Co-stimulatory receptors bind to B71, CD80, and B72, CD86 on APCs to provide secondary activation signals.
Okay. Types of cell-mediated immune responses. When it comes to the two types of T cell-mediated immune response, there are two that are discussed. CD4 plus T cell-mediated reactions, which is the delayed type, and hypersensitivity, DTH, and the CD8 plus T cell-mediated cytotoxic reactions. The CD4 plus T cell-mediated reaction mainly deals with the inflammatory response and tends to be more of a delayed speed, taking 2 to 4 days. This involves cytokines and such as IFN and TNF. While CD8 plus T cell mediation works with abnormal and dangerous cells, getting rid of them. This process is more rapid and happens within a few hours. It targets abnormal or dangerous cells, virus infected or cancerous cells. And it uses perform granzyme and the fast SL pathway.
Subclasses of CD4 plus and their role in cell-mediated immunity. CD4 plus T cells mediate cell-mediated immunity by differentiating into specialized subsets. Th1 cells activate macrophages and cytotoxic T cells to fight intracellular pathogens. Th2 cells promote antibody production and eosinophil activation against parasites. Th17 cells enhance neutrophil responses to extracellular microbes. The TFH cells assist B cells in antibody development. And regulatory T cells or TREGs suppress immune responses to maintain tolerance. These subsets work together to balance immunity and prevent excessive inflammation. Mechanisms of T cell mediated macrophage activation. T cells activate macrophages through classical M1 and alternative M2 pathways. The classical and alternative pathways is Th1 cells release the IFN gamma and engage CD40 and CD40L interactions to enhance macrophage antimicrobial activity. It leads to the production of reactive oxygen species, otherwise known as ROS, nitric oxide, and lysosomal enzymes for pathogen destruction. Th2 cells release IL-4 and IL-3, promoting tissue repair, fibrosis, and anti-inflammatory responses instead of microbial killing for alternative pathway. The macrophage responses for killing ingested microbes are ROS, which is produced by the NADPH oxidase to generate superoxide radicals, and hydrogen peroxide for pathogen destruction. Nitric oxide, created by inducible nitric oxide synthase to damage microbial DNA and proteins. Lysosomal enzymes, which aid in breaking down microbes after lysosome fusion. Pro-inflammatory cytokines, which is TNF alpha, IL-1, and IL-6, enhance immune responses. Autophagy and phagocytosis help eliminate intracellular pathogens and prevent their replication. Thank you. Okay. Okay, guys, I guess let me figure out if I can edit this right now.